All right, everybody, this is the answer key for the molecular genetics review worksheet. All right, and again, one thing I wanted to point out here, hopefully you noticed that the last 2 pages of this worksheet were actually the answers for majority of this worksheet here, okay, because I don't really care if you have the answers to this, right? My point is for reviews, is for you to be able to test yourself, see what you don't know, right? So I wanted you to be able to answer that worksheet and then be able to check yourself almost instantaneously. All right, so what this video's goal is to add extra information that maybe you didn't get, or to clarify some things that might have been a little bit more difficult here. Okay, so for this first question, where is the genetic information stored in eukaryotic cells? All right, well, genetic information we've been talking about is DNA, right? And that is stored in the nucleus, right? The DNA is stored in the nucleus and it never leaves the nucleus, right? That is one of the things we've really highlighted here. All right, for number 2, what materials compose genes? That would be the deoxyribonucleic acid. Again, that's the long form of the DNA, right? The shortened form is DNA. Long form would be deoxyribonucleic acid. For number 3, I didn't draw it here just for the sake of space, but there are basically 3 parts of a nucleotide, right? We've talked about this quite a few times. You've got your sugar, right? Whether it is the deoxyribose or the ribose sugar, you've got your sugar, you've got your phosphate group, and you've got your nitrogen base, all right? Now, there are 4 nucleotides that make up DNA, right? There are 4 nucleotides that make up DNA. We have adenine, thymine, guanine, and cytosine, right? And we've shortened them by just calling them A, T, G, and C, all right? Now, the shape of your DNA, right, when we're talking about it, again, we're talking about that double helix shape. You could also say twisted ladder, right? You could, multiple things you could say, but we've normally been calling it that double helix. Again, I would also accept a twisted ladder. For number 6, DNA has 2 strands. Do both strands go in the same direction? No, right? And again, I don't really care if we know a whole ton about this, but this is where we were talking about that 5' versus the 3', right? And those strands being anti-parallel. But basically what that means is they don't both go in the same direction. All right, for number 7, cytosine binds with guanine, right? We said chewing gum. That was our memory clue there. All right, and then in the process of copying DNA, we call that replication. We call that replication when we are making another copy of DNA. All right, for number 9, what molecule of heredity contains deoxyribose sugar? Shortened, again, DNA, right? I wanted to highlight that the reason we call it deoxyribonucleic acid is because it is made of a deoxyribose sugar. All right. For number 10, what kind of acids are DNA and RNA classified as? They are both nucleic acid. That's why they both end in Na. They nucleic acids. For number 11, what 5-carbon sugar is found in DNA? We just said it, right? Deoxyribose. For sugar that is found in RNA, that is ribose, right? That's one of those big differences between the two that we've talked about a few times. For number 13 here, in DNA, thymine binds with adenine, right? So our memory trick for that one was apple tree. So we had chewing gum, apple tree, right? For those 4 nitrogen bases, they could get into 2 categories, right? The pyrimidines or the purines. And we said the memory trick for this guy was the pyrimidines had the Y, so cytosine and thymine, because they both have Y in them. For purines, right? For purines, they don't have that Y, so that would be the guanine and adenine. All right, for 16, what type of weak bond holds the nucleotides together, right? So we're talking specifically about the hydrogen bond that holds them together, right? If we were to just talk in general what holds the 2 sides of DNA together, you could also say the nucleotide, right? But specifically, when we're talking about bonds, it is a hydrogen bond in that nucleotide, right? So you have a hydrogen bond that connects one side of the nucleotide on the DNA to the other side of DNA with that other nucleotide. All right, for number 17, what molecule carries out the instructions coded in DNA, right? Carries out, right, out of the nucleus. Okay, so it carries the instructions from the DNA out of the nucleus. That would be the RNA, right? And specifically, the type of RNA that carries the coded message, right? The coded message would be the mRNA. Remember, because M stands for messenger. All right, now, One of the reasons that RNA can do this is because it is made up of one strand, right? It is one strand, it is smaller, so it can go in and out of the nucleus. All right. For number 20 here, what type of RNA transfers amino acids to the ribosome for translation? Okay, so transfers amino acids. So amino acids are on the tRNA, right? The transfer RNA. So they ride in on the tRNA and they are linked together. All right, so transfer RNA brings those amino acids to the ribosome. All right, what molecule is made of ribonucleic acid? That would be RNA. Okay. For number 22, what is the process where DNA writes a piece of RNA, right? So going from DNA to RNA is known as transcription because it is transcribing or writing a copy, right? For 23, where are the proteins assembled in the cell? That would be your ribosome. It's in your ribosome. All right, and the ribosome is made up of that rRNA, right? Ribosomal RNA. For number 24, what do you call the decoding of RNA to link amino acids, right? So when we're linking amino acids to make a protein, we call that translation. That process is called translation. All right. What type of molecule usually ends in an ASE, right? We talked about two main ones, helicase and polymerase, and those are both enzymes. Now, typically, what enzyme unzips a piece of DNA into two strands? That would be helicase, right? And I want to highlight here, it's helicase when it completely causes hell or havoc, right? It's when it completely splits the DNA into two strands, it is helicase, right? Now for 27, which enzyme unzips a piece of DNA back into zips a piece of DNA back into one piece? That would be polymerase. Polymerase is also the enzyme that makes RNA, right? So polymerase has multiple roles here. All right. For number 28 here, what does a group of 3 nucleotides in a strand of mRNA form, right? So again, if we had GUA, we would call that a codon, right? Because it is the code for the amino acids that we are trying to link together. In RNA, what nucleotide replaces thymine? That would be uracil or U. Okay, for number 30, during transcription, what does a DNA molecule write its message on? That would be a piece of RNA, right? So it's transcribing a piece of RNA. For 31 here, what is produced from amino acids when mRNA and tRNA meet in the ribosome during translation, right? It's a protein, also known as a polypeptide, right? So either of those words are totally fine with me, okay? But basically, you have mRNA, it carries the message, tRNA starts linking together in the ribosome, and you make a protein. That is the end goal here. Now, for 32, what type of RNA has a codon of 3 letters? That would be the mRNA, right? So I want to highlight here, we've got codon and anticodon. So mRNA brings the codon. All right, tRNA brings the anticodon. 34, when do the codon and anticodons meet? In which process? Again, we're talking about it, it's translation, right? We are taking that message and now executing the creation of the proteins. All right, now for 35, this one was just kind of fun here. How many different combinations of A, C, G, and T exist? Right, it's 64, right? So I don't expect you to remember that off the top of your head here. Okay, basically what I was asking is if you take all of these different boxes, right, we have 4, 4, 4, 4, right? So you have 16 here, 16 times 4, right? That gets us our 64. All right, so that was our 64 there. All right, now, how many different codons make amino acids? Only 61 of those. Only 61 out of our 64. Because I want to highlight, there's these things called stop codons. We have 3 of them. And when you reach a stop codon, what that means is we stop making that protein, period. It shuts off, right? So we don't keep reading that code. We completely stop with that protein. All right, now, for 38, a stop codon stops the process of making, like we said, a protein or that polypeptide chain. All right, for 39, when does DNA replication take place? That is during the S phase, okay, during the S phase of mitosis. Now, this is not a huge point here, but I want to point out to you is that's what connects our mitosis and meiosis chapters to this DNA replication and to the protein, right? Without this S, without the synthesis, right, that's what that S stands for, without synthesizing or making these proteins, we will never be able to replicate a cell or to make a new cell. So we have to make sure that we can replicate the cells during interphase, specifically in the synthesis phase, before we prepare to split that cell into two cells or 4 cells. All right. What is protein synthesis? Again, it's just the process of making a protein. Synthesize means to make. All right. For 41 here, where do you find ribosomes in a cell? Floating in the cytoplasm, right? So we talked about that quite a few times, the cytoplasm, right? It is not in the nucleus. And this is something also that we didn't really talk about, but specifically it's attached to this ER or endoplasmic reticulum, right? That's just another part in the cell. What I want to highlight here is that, so these ribosomes are not just floating around freely in the cytoplasm. They are anchored somewhere, right? So it all takes place in very specific places. All right. Now for 42, which organelle does protein synthesis occur in, right? That would be your ribosome. Hint, it contains ribosomal RNA. For number 43, what makes up a polypeptide or a protein? Those would be those individual amino acids, right? We kept talking about them as like little links, okay? So you need multiple amino acids to make up a protein or a polypeptide chain. For number 44, give 3 examples of proteins in the human body. There's numerous, right? But 3 of my favorites here: hair, skin, nails. Okay, hair, skin, nails. You could have picked any others that you found online, and those would have been totally fine. Now, I just want to walk through one of these examples with you here, okay? Now, going from replication to transcription to translation to protein, okay? So this is one whole process here, and then this is multiple processes in order to have that protein synthesis, all right? So when we are replicating, we are just making a copy of the DNA, right? And we talked about this. We are using base pairs A, C, G, and T, all right? A, C, G, and T, and we just match those up, right? So if I do the first, 4 or 5 here, T matches with A, apple tree. So anytime I see a T, I write the A. Okay, for G, I have chewing gum, so I'm going to write my C's, chewing gum. All right, and then when I see T, when I see an A, I'm going to write my T's. All right, and for this one, it happens to have both of the same codes here, so that's kind of boring. I'll skip down and we'll do the last 3. So CCG becomes GGC. All right, now when we're going from replication, that's DNA to DNA. But when we're doing transcription and translation, we now start dealing with RNA, right? And with RNA, we no longer have U. We no longer have T. We have to use U. So our 4 nucleotide bases or nitrogen-containing bases are A, U, C, and G, right? So instead of putting a T, I'm going to put a U, right? If we go back here, for T, we still have A. For G, we still have C. But now, again, like we said, instead of A, you should have a U. Same thing. CCG becomes GGC. There's no difference there. The main difference is that we no longer have T, so we replace it with a U. All right? And good, we checked ACU. That's what we got. Perfect. GGC, that's what we got. Perfect. All right? Now, what we do is we use the mRNA to figure out what amino acid we're going to call. So ACU. If I scroll all the way down to where I have those codes, I said ACU. So ACU, it should be that guy. Okay? So we can abbreviate this however you want, THR, right? So ACU stand for THR. And THR is going to be attached to this tRNA, and it has an anticodon of, and that's what we got to figure out. A pairs with U, C pairs with G, and U pairs with A, right? And what you're going to notice here is it's almost identical to what the DNA we started with was, right? Again, just the only difference is instead of T, we had to put a U. All right, if we do our other one here, GGC, right? GGC, we got to figure out what codon we're calling. So we've got GGC, we've got glycine. Okay, GGC, we've got glycine. So if I scroll back up here, GLY. All right, and GLY is going to be on the tRNA with the anticodon of CCG, right? And since we didn't have any A's or T's or U's, right, we had no change here. So CCG for the tRNA is exactly the same as the CCG for the DNA. All right. Now, if you want the rest of the answers to that, you can obviously look at the bottom of the actual assignment here. All right. And again, what if you should have seen, I did put in the bottom of my copy, I did put the notes that I asked you to go over. So again, please take a minute to review. It has some really, really good information and really good pictures in it. All right. Again, hopefully you found some value in this. There was really no major things you had to accomplish for the Nearpod. The Nearpod was solely for you and to help you out. Again, other last concluding mark here is that on Classroom I did make a new post, right? So it has it's just a material post, and it has all the videos that we used, including the videos in the Nearpod that we helped use to explain some things that were going on in Chapter 12. So all of those videos are posted in one link, and they are available to you. So please feel free to review them as you need them. All right. If you've got any questions, as always, you can reach out, and we'll start class with answering those questions.